Okay, so now what I'd like to do is I'd like to talk about the neuron at rest, which is going to be this resting membrane potential. Make sure you look at this learning goal here after we've talked about it. You want to make sure you understand all of this information. Don't skip any part. So let's just begin again by reminding you that we're talking about this image here, what the nerve is like, our neuron is like at rest. And so we're going to be looking right here. Okay, now this here is a close-up of the membrane of that neuron. Okay, so you may recognize the phospholipids, the heads, the tails on either side. Over on this side is extracellular fluid. Extra means outside the cell, so it's the fluid outside the cell. And over here is going to be the cytosol, so this is inside the cell. Okay, now to understand these figures in the books, you need to make sure that you look at the key. Okay, so this shows here that all the purple dots are sodium ions. All the orange dots are potassium. All the green dots are going to be chloride. Okay, and then these big purple ones here are labeled as a protein. You notice they all have charges too. We either have positive charges or negative charges. Okay, to understand a neuron at rest, we need to understand where all of these parts are located relative to the membrane. So first let's start with sodium. So you may notice you've got one sodium here, but most of the sodium is on the outside of the cell. That's important. So at rest, high concentration of sodium outside the cell. Now, potassium is going to be just the opposite. Potassium here, you notice, is mostly on the inside of the cell. You might have a couple on the outside, but mostly on the inside. Now, chlorine looks pretty even on both sides, but what you should notice that's present on the inside, but not on the outside, are all of these proteins that are negatively charged. Okay? And so what that means is overall, the charge on the outside of the membrane is positive, and that's what these positive signs are representing. While the charge on the inside of the membrane is mostly negative. Okay, now let's look at what's going on with the movement of these ions. These arrows here are showing that the ions are moving. And so this little blob stuck in the membrane is a sodium leak channel. If you need to review channels, go to chapter 3. But know that a channel is passive transport. It moves things from high to a low concentration. And it's a sodium leak channel, so it's only going to let sodium through. And so that's what this is showing. Sodium moves passively through the leak channel. But then we can see over here, the pump will take it and move it back to the outside. So this ion is continually circulating passively through the leak channel and passively through the leak channel and actively through the pump. Now, potassium is going to go the opposite direction. Potassium starts out high levels inside the cell. And so it will passively move through the leak channel. and then actively be pumped back to the inside of the cell. Okay, now I'm going to show you an active learning exercise where you can practice moving these ions at home to make sure that you really understand the concept. Okay, so this is showing the printouts from the active learning exercise that you can print off at home. I don't have all of the pieces out here. I've got the sodium leak channel, I've got the potassium leak channel, I've got the sodium exchange pump, I've got a whole bunch of sodium ions, and I've got all of my potassium ions here in orange. The other two pieces I have out are negative membrane potential and positive membrane potential. So I'm actually going to start by moving these for a minute, and the first thing that I want to do is I want to draw the cell membrane of the neuron. Okay, so this is the cell membrane of the neuron. Okay, now I'd like to make sure that I label the outside of the cell as well as the inside of the cell. Okay? Now what we're going to do is we're going to place each of these channels and pumps in the membrane. So there's the exchange pump, here's the potassium leak pump, here's the sodium. Then I want to make sure that we start off with everything on the right side, or on the correct side. So first we're going to put all of our sodium on the outside of the cell. So we have high levels of sodium outside to begin with. Okay? Then we're going to put potassium, most of it, on the inside. And that's because we start with high levels of potassium inside of the cell. The other thing we have is that we need to make sure we know what's positive and negative. So positive, and I'll write it here because I know you can't see very well on the video, is located there on the outside, and it's generally negative on the inside. In fact, you should know that they've measured how negative it is. It's actually minus 70 millivolts. Little m and big V stand for millivolts. Okay, so now let's show how these things move. Okay, so first of all, let's start with sodium. The leak channel moves things from high to low, so sodium first moves from outside to inside. It will then be taken by the pump and moved from inside to outside. So again, it goes passively inside the cell, actively outside. So we actually have it constantly moving in a circular pattern in the cell. Okay. Now, this is sodium, and so potassium is going to be the opposite. Potassium passively moves out of the cell and then is pumped actively into. Okay? And so again, potassium moves passively out, and then the pump takes it and will pump it back in. You have to have this movement of ions to get a current, which gives you the minus 70. If these things stop moving, you don't have that same condition exist in the cell.